Hi, everyone, and welcome to People Behind the Science. I'm Dr. Marie McNeely, and I appreciate you joining me today for episode 783 with our guest, Dr. Ruchira Singh. Listeners, if you like this episode and you want to check out more stories from amazing people doing great science today, you can find more episodes of our show on Apple Podcasts, Spotify, or wherever else you like to listen. And today, Ruchira has some excellent stories to share with all of us. So get ready to meet another one of our brilliant people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am looking forward to speaking with our guest scientist, Dr. Ruchira Singh. So, Ruchira, welcome to the show today. How are you?
I'm doing good. Thank you. Thank you for having me.
Well, thank you so much for joining us, and I'm excited to learn more about you and more about the wonderful work that you're doing. But before we get into those details, let me first take a moment to tell our listeners a little bit more about who you are and how you got to where you are today. So listeners, Ruchira is an associate professor and the recipient of the Dean's Professorship in the Department of Ophthalmology at the University of Rochester Medical Center, where she also has joint appointments in the Department of Biomedical Genetics and the Center for Visual Science. She received a bachelor's degree in computer science from JNTU in India, and she completed her graduate studies in physiology at Kansas State University. Next, Ruchira conducted postdoctoral research in pharmacology at Yale School of Medicine, followed by a postdoc position in stem cell research at the University of Wisconsin-Madison. She joined the faculty there at the University of Rochester in 2014. And Ruchira, today we are excited to get to know you as a scientist, of course, but also as a person. So can you start by telling us, what do you like to do when you're not doing science?
I truly enjoy spending time with my kids. I have a 6-year-old son and an 8-year-old daughter, and they both give me so much joy. We don't do anything in particular, but small things bring so much joy to them, like going to McDonald's or the park. Mm-hmm. Our favorite place to go to is the kids section of our local public library, where my son plays, my daughter browses books, and I catch up on my emails and work all at the same time.
That sounds amazing. I think spending time with family can be a great way to unwind after a busy day in the lab. And I know you're doing some amazing work in the lab, and I'd love to touch on that as well. So, Ruchira, how do you describe what you do to someone who's perhaps outside of science or maybe just not familiar with your specific field?
So I mostly, again, this is talking to like students in my daughter's school and stuff. Basically, I tell them I'm a scientist that works on the eyes. I try to understand why some people and some kids cannot see as well as others and use that knowledge to develop treatments for specific eye diseases like age-related macular degeneration. To do this, I take skin or blood sample from people that either can see well or those that cannot see so well and have vision problems. I convert them into stem cells and then use them to make cells from the eyes that we then study in the laboratory. By making eye cells from people that have the disease and those that don't have the disease allows us to compare what are the differences between these cell types. And this knowledge allows us to understand more about why some people cannot see as well as the others. And this is important for developing treatments for eye diseases.
Oh, that makes sense. And I know age-related macular degeneration is a really common form of vision loss or common cause of vision loss. So for people who aren't familiar with what this term means, could you describe it briefly?
So age-related macular degeneration is age-related irreversible blindness. So basically the part of your retina, the central retina, the macula, the cells in it, the photoreceptors, the light-sensing cells stop working. And so that reduces your ability to be able to see. And what happens is your central vision is the first one that goes away at the first side of the disease. And even before the disease develops, there is these early pathological hallmarks called drusen that develop. But over time, there is progressive loss of central vision. And then basically it leads to irreversible blindness. And 54% of all the blindness in the United States is because of age-related macular degeneration.
I think that's remarkable and a very important area of study here. And I know just being able to work on a condition like age-related macular degeneration and other forms of vision loss can be inherently motivating because it has the potential to have a big impact on people's lives. But let's take a moment to talk about motivation. I think for me, at least, having a little quote or a saying or some mantra in my mind as I'm going through a tough day can really be helpful. So for our listeners out there who might need a spark of motivation, do you have a favorite quote or a saying that really resonates with you?
There are a lot of tough times. So I'm always trying to drive myself to go past things. So I tell myself, tough times don't last. Tough people do. That's my favorite one. The other thing I keep telling myself is this too shall pass. Every time I have a difficulty.
So I know becoming a tough person isn't easy. And I think oftentimes mentors, role models, people you meet throughout your life and your career can kind of help shape you, help you build that thick skin. So Ruchira, when you look back at your own career and your own life, are there particular people who rise to the top of your mind as people who really had an impact on you?
Yes. So actually, my PhD advisor, Dr. Feline Wangerman, she's probably why I am in science. And she was the most determined, focused, and careful scientist I've ever known. She once said to me, I don't do anything I cannot give 100% to. And when I was a graduate student, I did not like that at all. Now that I look back to it, I think that's the best advice I've ever gotten, is that if you really want to do something, you want to give it your best. And I think she taught me the rigor and respect for science. So I'll always be grateful for that.
Definitely. And I think in science, the pressure can be high, and this feeling of having to give 100% to everything can be difficult when you feel like you're juggling just a lot of things. So how do you balance that, Ruchira?
I learned this again from another mentor. I actually met this lady once in my life, but she told me that your life outside work is as important. And no matter what, 5:30 to 7:30 of my day always belongs to my kids. And I make it a priority to be with them. And I think no matter what happens, how bad the day is, whatever it is, just being with them makes it all fine at the end of the day.
Definitely. I think setting those boundaries and being firm on them is difficult, but it can really make a difference in your life when it's so easy, I think, for science to just swallow up all of your time.
Exactly.
Well, let's talk about your journey then next, Ruchira. Can you share with us how you first got interested in science and even started on this pathway?
So I did not take a traditional route to science at all. So my father is a doctor in India. We were 3 kids growing up, my brother, my sister, and me. Both my older siblings had not decided to pursue science at all. So my dad was hoping I would be the doctor. The first chance I got to be out of it, I decided I'm going to leave biology. So it was 1995 and I was in high school, and there was this new craze about computer science. And my school had come up with you could leave biology and choose computer science as a major. And I was like, okay, great. I'm doing this. So I don't have biology anymore. I don't have the pressure from the family anymore. And I was pretty happy with it. But then the very same year, I actually got hit by a bus. I came under a bus. I had a major accident and I was in the hospital for several months. And I saw a lot of people suffer through it. I was lucky that my dad had the resources, and one of his close friends was a surgeon who saved my leg. And I honestly, at that point when I was in the hospital, I really wanted to go back to science, but I was in India. And in India, it's not a norm to change your profession. And I did not know what to do. So I continued with computer science and I became a computer science engineer and did my computer science engineering from a pretty mediocre college in India. And all my friends were moving to the United States to do their masters in computer science. Some were getting married. So I just followed the direction and I came to the US to do my master's in computer science at Kansas State University. And I was trying to make some money on the side. So that's how I met my PhD advisor. I was designing her webpage. And they were working on hearing loss in a mouse model. And I would keep asking them questions about how do you do this and how do you do that? And I was very intrigued by all the research around me and all this neat equipment and experiments that the people were doing that I had no idea that you could actually be involved in all this. And one day out of the blue, Dr. Wangerman asked me if I wanted to be more involved in the research in the lab. And I told her, of course, but I have no background in biology. So she gave me a few books and she said, start reading and come up with a project idea. She would ask me questions about what I was reading and stuff. And I truly enjoyed it. And so she once told me you need to present to the lab about gap junctions. So that was the first presentation I did about gap junctions. And I truly enjoyed it. And at the end of the presentation, she came up to me and she said, have you thought of science as a career? I was pleasantly very surprised, but I did not believe that this was possible with my background. But under her mentorship, I took several prerequisite undergraduate courses, and then I got enrolled in PhD in a program in physiology. And after completing that, I moved to Yale School of Medicine to do my postdoc in pharmacology, where my focus was diabetic neuropathy. And again, at that time, I got married and I had to move to Wisconsin-Madison. And I quickly found the lab of Dr. David Gam, who's a retina clinician who works on patient-derived stem cell models of retinal diseases. And that's where I started working on stem cells in the eye. I finished 3 years of my postdoc with him and moved to Rochester in 2014 as an assistant professor of ophthalmology. And now I have been here for 10 years, still working on eye and stem cells.
Well, Ruchira, you have an amazing story, and I'm very curious. I think a lot of science has moved towards more computational and mathematic approaches. Do you find that having this background in computer science has actually maybe made you a better scientist?
I feel like I look at things a little differently than if I was a hardcore trained biologist. I don't necessarily as much use computational tools anymore in my research, but I do know where to find those resources when I need them.
So then you mentioned you've been there now at the University of Rochester for some time. Your lab is doing some really cool work, and I'd love to dive into the details of one of your projects. So is there one ongoing at the moment, Ruchira, that you are just the most excited about and want to share with us today?
Yes. So my laboratory uses several different models to study retinal degenerative diseases or eye diseases. And we do imaging on patient retina, and we use patient-derived stem cells, and we use animal models when available to study the mechanisms. The one project that I'm truly enthusiastic about right now is actually on Batten disease, which is caused by mutations in CLM3 gene. This is a neurological disorder, but there is vision loss. And surprisingly, clinically, the first symptom that precedes neurological symptoms by several years can be vision loss. So when I started working on this disease, the consensus in the field was that vision loss in Batten disease is because the neurons in the retina are getting lost, just like the neurons everywhere in the brain. I remember that the first time I looked at a postmortem tissue of this disease, I was struck by the changes I saw in a non-neuronal cell type, the retinal pigment epithelium. Given what I knew about the retina and how the retina functions and the importance of this particular cell type for retinal cell health, I was like, we need to study this non-neuronal cell type in this neurological disorder to understand why vision loss is happening. Since then, we've published that retinal pigment epithelium function, specifically phagocytosis of shed outer segments, which is an essential function for photoreceptor or light-sensing photoreceptor cells. Their health is impaired in CLM3 Batten disease. And now we are using stem cell models and a large animal model of CLM3 disease to investigate exactly how RPE cell loss or retinal pigment epithelial dysfunction is contributing to vision loss in CLM3 disease. And perhaps this can actually lead to development of rational therapies targeting the early vision loss in this debilitating blinding disease.
Very interesting. And for folks out there who might not have heard of Batten disease, can you just briefly describe what that might look like?
It's a group of disorders, right? So I study Batten disease, which is caused by mutations in CLM3 disease. So there are several different forms of it. And these are basically lysosomal storage disorders. They affect mostly young kids and they lead to neurological dysfunction. And in a lot of these diseases, vision loss is the first phenotype, but ultimately over time, there's a lot of neurological impairment. And ultimately, the most common form, the one that I study, the CLM3 disease, a lot of people live up to their mid-20s or 30s, and it's a fatal early onset childhood neurodegenerative disorder. So it's one of the diseases that I am most invested in studying, and I want to understand how it happens and if I can make a difference.
Absolutely. And I think that the work that you're doing to better understand the mechanisms could really potentially pave the way for new therapies, because it sounds like there aren't currently therapies to really address this debilitating and fatal condition.
No, there are no. This is indeed a very important area of research. And there are right now a few people that are trying some clinical trials and there's some therapeutic avenues, but there is very limited understanding of how the disease happens. Now, there is avenues for gene therapy and stuff that is still, again, not out there for CLM3 disease yet, but understanding how the disease develops is very important for developing rational therapies.
Well, that makes sense. And I appreciate you sharing one of these exciting research projects that you're working on in your lab. And I think doing this kind of research can have its ups and downs. And I think in science, there are a lot of downs, a lot of failures, a lot of struggles that you have to work through to kind of get to these big breakthrough discoveries. So do you have an example of a time where you really struggled with something, or perhaps you had a major failure in science? And if so, could you maybe walk us through how you got through that tough time?
Yeah, I don't know if it was a major failure in science, but getting my first R01, which are these prestigious National Institute of Health grants that are deemed to be very, very important for getting funding, and as a success metric in academia, was a total nightmare. I was getting a lot of recognition and funding from private foundations, but they're not as substantial as NIH funding. And I was finding it very difficult to convince the NIH study section reviewers that the model system that I was proposing to use, which is patient-derived stem cells, was a good platform to study retinal diseases like age-related macular degeneration. And this was very challenging. I submitted that grant several times and I kept getting the same feedback. This is an age-related disease. This cannot be studied in a cell culture model. You need an animal model to study this disease. And at that point, my choices were either that I just switched the focus of what I was doing, or I showcased why this model can be used to do this. And luckily for me, we looked, and we were able to figure out that in the stem cell derived model, when you have patient derived cells, you can mimic key aspects of the human disease pathology, like drusen, that had not been able to be done in an in vivo or in an animal model previously. So we published that paper in PNAS, and that opened the door for this line of investigation and for my R01 to get funded. But it was a very stressful time. But I had to make sure that I kept working on it to get that convincing data to open up people's thought process on being able to use patient-derived stem cells as a relevant model system.
I think that perseverance paid off, it sounds like. And for you, I mean, there has to be moments during all of this where perhaps you question whether this model is actually the best choice. Were there those moments for you? Were there moments where you thought about giving up and maybe pivoting to those animal models or pivoting to a different area altogether?
Of course, because it is very important to have funding to support your work. And a lot of science requires money to be able to do what you're trying to do. And I was like, yes, I really want to do this, but if I don't have the funding to do this, there's no way this is going to be possible. So I was actually thinking about, yes, moving into an animal model, but I wasn't ever convinced that in isolation, an animal model would be sufficient to study a human-specific disease. And now coming back a circle, we've come to a place where we use both stem cell-derived models and animal models together as complementary approaches, because there are aspects of things, for example, toxicity, that you can only test in an animal model where the whole complexity of a living being is there, right? So we've now integrated them, but as a starting point, I'm still glad that we started with using patient-derived cells, because I think that gave us some important insights into the disease mechanism that we would not have gotten otherwise.
Definitely. And it sounds like this challenge had a happy ending. You did ultimately get funding, which I think is wonderful. And I love to celebrate these success stories in science, because I think often there's so much work and difficulty getting to that point of success. So is there another success story you'd like to highlight from your career, Ruchira, whether it was a big success or even just a small but particularly meaningful one for you?
Yes. So again, like coming back to this whole animal model and stem cell thing, right? So as I said, when I embarked on studying the molecular basis of age-related macular degeneration, I tried to use the patient-derived cell model, and I had a lot of challenges in trying to convince people that the human cell model, although it is a cell model, is an important model to study the disease. My rationale was very simple. My rationale was like, mice don't develop the disease. You cannot study a disease in a model system that does not develop the disease-associated pathology. And I agree that patient-derived cells are not in vivo and they cannot capture the complexity of the human disease in a cell culture model. But I was really wanting to pursue using a human cell model to study macular degeneration. And interestingly, when we were dissecting the process of how this is happening, we were able to, as I said, capture the early pathological hallmark of age-related macular degeneration in a dish, the drusen deposits. We were then trying to figure out how were these drusen deposits forming in the patient-derived cell models. And we discovered that an enzyme that is crucial for drusen formation in the human cell model is actually not functional or is functionally absent in certain strains of mice that have been routinely utilized to develop animal models of macular degeneration. And this told us that maybe this is why mice don't develop macular degeneration. So being able to link that, the protein that was playing an important role in my patient-derived human cell model for developing the human disease phenotype is actually not present in the mouse models that are being used to study macular degeneration, to me was a very big win. I was like, okay, finally, I have convinced myself as to why this approach was needed. And now we have understanding of likely why mouse models of macular degeneration do not develop the human pathology.
I think that is fascinating. I think these aha moments in science are sort of rare in a scientist's career, but it sounds like this one was a clear moment where you saw, all right, this is what's happening. This is the mechanism behind it. And this is clearly absent in mice. So how did you celebrate this big breakthrough?
I think a lot of people in my lab were just so excited that this was indeed true. And we just went out and we celebrated, had lunch. And I think we still keep talking about it. We're like, oh, wow. So we were able to link the stem cell model and figure out why maybe it's not happening in mice. We're still trying to figure that out if that's really true, because we're working on the mice to now see if we now introduce this enzyme, are we able to get the disease? So I think the celebration was basically that aha moment. And I think not just me, the whole lab, we were so happy that after all, there was a rationale for why we use a human cell model.
Definitely. And I think celebrating these successes with the group is wonderful because oftentimes there are so many people who contribute to different pieces of these projects over years that kind of culminate in this big moment. And I think working in the lab, coming to these big breakthroughs can really motivate you to go all in. Like you said, you just want to keep talking about it and kind of figuring out these next steps and next pieces of it. But we like to encourage our listeners to take a break, to expand their minds and to read broadly. So we love talking about book recommendations. So Ruchira, do you have a favorite book that you'd like to recommend to all of us, whether it's related to science or not?
So I actually don't read a lot of books in science. Mm-hmm. I read a lot of publications that are very focused on the area of research I do, but I was a very avid reader growing up. And the books that I like are, again, I don't know if that's a good recommendation for this show, but I grew up in India. And one of my favorite books is Malgudi Days. It's actually a collection of short stories in a small Indian town. And it basically captures the spirit of life and relationships and its complexity. But it also has a deeper sense or deeper meaning that actions of most people ultimately have a well-meaning purpose. And it's a very funny book, but it has a very strong implications in terms of life and relationships and our understanding of it. I truly enjoyed that book.
Well, this is a wonderful recommendation. Well, listeners, we will add this to our book list on our website for everybody. And this is a wonderful recommendation. And I think as a scientist, we've highlighted that throughout your career, you've actually traveled quite a bit, even just at these different career stages that you've gone through from India to the United States at various institutions. And I think as a scientist, travel is a big part of life, whether it's for work in these changing positions along your career trajectory or for conferences, working with collaborators. So, Ruchira, do you have a favorite place that your science has taken you over the years?
I don't know if I gained very much scientifically from this particular trip, because again, these same conferences happen in different locations. But the favorite place that I've ever been to has been Gold Coast, Australia. It was just refreshing with all the beaches. I really enjoyed the fresh bread and cookies. And it was the first time I got to hold a koala.
Oh,
I think it was just memorable.
Well, that sounds wonderful. What was the occasion that brought you to Australia? Was it a conference?
It was a conference, yeah. I hate traveling, so I never travel unless I have to go for a conference.
Well, did the conference go well?
Yeah, I mean, I truly enjoy conferences because you learn so much about what people are doing, what's groundbreaking, what's going to be coming up soon, what new approaches. So I really enjoy going to scientific conferences. I wish I didn't have to travel to do that.
Were there particular things that you saw or did in addition to holding koalas, seeing these beaches that really made the trip particularly memorable?
I find water very relaxing. And just being able to be there and observe. And again, like it was just how nice people were, how accommodating to tourists like me, and how genuine the whole place was. The times that I was not at the conference, I was probably just sitting somewhere and watching water. And it was just very beautiful. And nature has this very calming and relaxing effect on me.
Well, I think that sounds amazing and nice and serene, which I think is good. Sort of a break from the chaos of a conference.
Yes.
And I know over the course of our conversation, we've talked about some of your lab members and mentors and folks that you've interacted with over the course of your career. And I think these people are what makes science great. I think oftentimes, unfortunately, scientists are portrayed a certain way in the media with their sort of stiff white lab coats and very serious demeanors. But I think so many people I've met in science are these funny, fun, amazing people. So do you have an example that showcases this human side of science, Ruchira, whether it was a quirky tradition or a funny memory or something else from the lab that you've experienced?
I'll talk about a funny experience in a second, but I think over the years, I've learned that a lot of people that I've worked with in the lab have been very, very helpful in terms of every time I've joined a new lab or been in a new place, the human side of it, right? There have been times when people have had to take up a lot of time away from what they were doing to be able to teach me something new. And I've always appreciated how throughout my career, people have taken that time to invest in me without any expectation of what they would get back. So to me, I feel like scientists as a group, right, we are very motivated by finding what we need for improvement of maybe the health or whatever. Like we are motivated to find the cellular mechanism, the cure, the whatever. And we work as a group and we are like very, very invested and appreciative of each other's effort. And we go beyond. In every lab, I can probably count 2 or 3 people that were very instrumental in me being able to publish or achieve or learn anything. So having said that, my funniest memory in science is actually gross. And I'm not going to name the lab or person that was involved. But a colleague of mine was holding a mice in her hand and she was trying to point out some important detail about the experiment we were doing and whatever. And she was so engrossed in figuring this out that the next thing we knew, the mice had peed in her mouth.
Oh, no.
And it felt bad for her. But now when I look back at it, and that's one of the memories I cannot forget. And I'm like, oh my gosh, she was so invested in it.
It just shows the mishaps I think that can happen in labs sometimes too. Well, I think some of these traits that you highlighted as well, this generosity, this solidarity are things that I've seen in science as well. And I think, like you said, it is this sort of group working together towards these big picture goals. And I think your lab is working towards a really important big picture goal as well with finding out the mechanisms and then perhaps ultimately working towards cures for some of these eye diseases. So if we were to take away the things that normally hold you back, Ruchira, whether that's funding, staff, technology, time, feasibility of projects, whatever the case may be, and you could do anything. And it doesn't even necessarily have to be in your field of research currently, but do you have one question that you would want to answer most?
I do. So as I said, I'm like really invested in lysosomal storage disorders because they seem to be affecting young kids. So understanding why neurons degenerate in these lysosomal storage disorders like Batten disease. Right now, we just study the CLN Batten disease caused due to mutations in CLN3 gene. If I could have my way, I would just put my whole lab to studying the different forms of Batten disease and other lysosomal disorders and trying to understand if there is parallels or interconnected pathways or how in these umbrella of diseases, how is vision loss occurring? And understanding vision loss, I think, will have broader applicability just than vision because it will help us understand in a broader context how the disease is developing and how neurons are getting to degenerate. So I think if I could have it my way, then I would just put all the resources I have, use human imaging, use stem cells, use animal models, everything together to study as much about lysosomal storage disorders as I could.
Well, this sounds amazing. I love this idea of being able to go all in if there were no barriers. I think this is a tremendously important area of research, and we appreciate the work that you're doing in this area. And I think to be able to do amazing work in science, sometimes you need that key piece of advice at that right moment along your career pathway. So for listeners out there who might be at a fork in the road or just working their way through their own journeys, do you have a piece of advice that you could share, Ruchira, that really helped you along your own journey?
I mentioned one of it already. My PhD advisor had told me that give 100% to whatever you do. But I think one piece of advice that, again, I can maybe kind of briefly mention it was that I met this professor by the name Dr. Rosalie Crouch, and I only met her once. And she has had a great influence on me as a scientist. She'd done some really awe-inspiring work on improving our understanding of retinal biochemistry and stuff. But when I met her, I was actually interviewing for a faculty position. And I asked her, is there a piece of advice? You're so successful. And she said, there's one thing you can do right is try to find time to do something other than science. And she said, if you have kids, for example, with her, she mentioned that she always found time for her kids every day. And she made it a point to be with them for dinner and before bed, even if that meant she had to go back to work. And I think I took it to heart. And at that moment that she told me this, I did not have kids. I was struggling with infertility. But when I did have kids, I always try to take time away from science, because sometimes when you step back, you think better. And at the end of the day is my de-stressor. But I think the important thing is to be able to not just make that the only focus of your life and have something else to lean back on.
Definitely. And I think this advice can mean so much when you're getting it from someone who's been through it, who has been successful in the career, and almost gives you permission to feel like you can stop at the end of the day and take that breath and take that moment with your kids.
Yeah, I think that's absolutely important.
Well, we appreciate you sharing that advice. And Ruchira, is there any other last piece of advice from you, or maybe a last piece of inspiration that you'd like to leave listeners with at the end of our conversation today?
What I've learned from my journey thus far is to learn from people around you. And I think what I've benefited the most has been from seeing how my mentors and other successful scientists approach science and the effort that they put into finding answers. And there's no shortcut. It takes a really extensive time. It takes a lot of investigation. It takes a lot of work. And I said before that the two very different outlooks, inspiration that have formed the framework of my life have been, one was my PhD advisor's outlook, is to always give 100% to everything you do. And the second one was Dr. Rosalie Krause's advice that you need to find time to do something away from science every day. In my case, it's my kids. So also prioritizing life outside of work is important.
Definitely. I think when you're at work, you give 100% to work. And when you're at home, you give 100% to home. And I love that balance that you've established for yourself.
Yes. And time for yourself. I'm not good at that one.
Yeah. Well, we appreciate you sharing this message with all of us today. If our listeners would like to learn more about you, Ruchira, and the work that you're doing, what is the best way for them to do so?
You can go to PubMed and you can search my name, Ruchira Singh, but I have a very common name. So you might have to search for Ruchira Singh in Rochester. On PubMed.gov, you can go to my lab's website at the University of Rochester. And if you really need to reach me or have a specific question, you can reach me via email. And that's again listed on my website.
Perfect. Well, we appreciate you sharing those resources. Listeners, definitely take a moment to learn more about Ruchira, learn more about the amazing work that she and her lab members are doing. And Ruchira, it's been such a pleasure to have you on the show today. Thank you so much for your time.
Of course. Yeah, no, thank you so much for having me. And I am truly grateful for the listeners who would be listening to my experience outside the science. And if anybody has any questions or would like to reach out to me, please feel free to email me or check out more of my work at PubMed and stuff.
Perfect. Well, thank you again for your time. And listeners, we were delighted to have you here with us as well. We hope you'll join us again next time for another episode of People Behind the Science.